Hello guys, good afternoon. Thank you for joining us again. So this session is GCSE Biology with a focus on infection and response infection, and we've got some exam techniques that are going to be shared as well. I know that some of you may have tuned in for physics, however, we've had to do a swap. So physics is going to be hosted at 3:15 this afternoon instead. So if you want to catch physics, you can come back at 3:15. Otherwise, we've got some biology here, and it's going to be delivered by the amazing Charlotte. Charlotte studied at University of Exeter and has taught many lessons here at my tutor. So I'm sure that you guys are going to have an amazing lesson. For those who are joining us for the first time, the structure of the session is as follows. So we have 45 minutes. It's normally kind of dedicated to content delivery time with some 15 minutes at the end for Q&A with Charlotte. So the total session length is going to be an hour long. Throughout the session, you can pop any questions you may have within the chat function, or you can also put the questions within the Q&A. Both are at the bottom of your screen. So do please let us know if there's any questions that you have. All right, if you're ready, Charlotte, I'm going to leave it to you to take it away.
Okay, thank you. So let's move to my tutor whiteboard there. So apologies for the abrupt change in timetable. So I normally take a biology class at 3:15 on the timetable, but today we had to do a swap. So I'm doing my class now, and then physics will be at 3:15 as said. I'm just going to load up Q&As now so that I've got that there. So this session is about working through kind of a summary of infection and response, which is what we've been looking at up until now. We've been talking a lot about diseases, a lot about different types of diseases, and also how we can treat and prevent diseases as well. So very topical at the moment while we're all at home. And today we're going to look at the sort of questions that you might get on this topic in an exam and how to answer them properly. So I'm going to show you some techniques for doing that, and we're going to kind of treat it as a bit of a revision session as well. I've got a plan to do some sort of quizzes and bits and pieces like that. So what I'm going to say is that if you can make sure that you know where the question and answer function is on your toolbar, that's where we're going to put any questions about the session. And then we're going to use the chat function as where we can put in any answers to quiz questions and things. That should keep them separate and mean that I can make sure that I can answer all of your questions without missing any as well. So I've got both the chat and the Q&A up so I can see. And let's give it a go, see how we do. So let's have a recap of kind of the infection and response module, what that includes and what we've gone over in just the last couple of sessions. So we looked at communicable or infectious diseases. So we talked about what those are. So diseases that can be passed between humans or between humans and organisms, so animals as well. We talked about what diseases and what pathogens are. So pathogens are microorganisms that cause disease. So not all microorganisms are pathogens, but pathogens are microorganisms. So for example, we have gut bacteria that are really good, that help us with our digestion. They're not pathogens, they don't cause disease. Whereas other bacteria do cause a lot of diseases in the body and they are pathogens. We talked about, let's clear up the difference between pathogen and disease first. So pathogens are the microorganisms that cause the disease, and the disease is the collection of reactions in our body as a response to that microorganism. So we talked about viral diseases, we talked about bacterial diseases, fungal diseases, and protist diseases. So 4 different types of microorganisms that cause disease that are pathogens. And we talked about a couple of different examples of each of those that we're going to run through in a bit. And then we also talked about how we can cure, treat and prevent disease. So we talked about our human defense systems, our skin, our saliva, our tears, stomach acid, and we talked about white blood cells and our immune system. And then we also worked through vaccination, how that works, why it's important that we have vaccination and how it can lead to herd immunity. We talked about antibiotics and painkillers and how overuse of antibiotics can lead to antibiotic resistance. And then we talked about how we discover and develop drugs, so the types of testing that take place. There was also a session yesterday covering monoclonal antibodies and some of plant disease, which is part of the biology only and higher, biology higher level. So we're not going to concentrate too much on that today, but more on exam technique. So I thought we'd start with a quiz. You don't by any means have to get all of these right. I don't mark them or anything, but I thought it'd be quite a good way to start. So I'm going to put up the name of a disease, and just had a chat question come in. Are you covering the same topics as the last webinar? Are you going to go in more depth? So we're covering similar topics, but we're looking at the sort of questions that you get about them in an exam, rather than the topics themselves. So we won't be going into as much depth as we have before. We're just going to look at how you would answer a question about them. Okay, so quick quiz. Is this pathogen a fungi, a protist, bacteria, or virus? Answer in the chat. HIV, which is human immunosuppressive virus. Very good, lots of virus coming in. So the V in HIV does stand for virus. I suppose I should have put really HIV and AIDS, because HIV leads to AIDS, which is a disease of our immune system, makes us vulnerable to other things. Good, loads of people saying virus. Use the chat function if you can, rather than the question and answer, and we'll try and create the question and answer function for questions about the content. Okay, how about, oh, excuse my terrible spelling, gonorrhea. Oh dear. There we go. So we talked about gonorrhea. Gonorrhea is an STD. It's passed on through sexual intercourse. Is that a bacterial pathogen, a protist pathogen, a virus, or is it fungal? Good, lots of people saying bacteria. That's completely right. So gonorrhea is a bacterial disease. Okay, the next one. So let's talk about so I put 2 words here. First one is the organism that causes the disease, and then malaria is the disease itself. Is that caused by a fungi, protist, bacteria, or virus? Protist. Very good. Fantastic, yes, protist is correct. So protists are a funny group of organisms that don't really fall into any category. They have some fungal qualities, some bacterial, and some viral properties. They are eukaryotes, so they have a nucleus, unlike bacteria, which are all prokaryotes. And Plasmodium is an example of those. There's another one, rose black spot disease. That's a disease that we get in plants, causes black spots all over the leaves, stops the chlorophyll from working properly and stops the plant from growing properly because it can't absorb that energy from the sun. Good, yes, that is a fungal disease. Well done everyone that's written fungi, fantastic. Another one, next one down, athlete's foot. Remember there was a terrible picture of athlete's foot that I posted. It's a disease that can affect your feet in rare cases. Is it caused by fungi, protist, bacteria, or virus? Good, lots of people saying, not so sure about this one. Thought anyone that was in the session would remember this one. It was a terrible picture. It looks really, really awful. The correct answer is fungal. Athlete's foot is caused by a fungal organism. So well done those who said fungi, some of you said bacteria. That's kind of an automatic reaction to think of, that when something looks really horrible, which athlete's foot does, people often think, oh, must be bacteria. But actually athlete's foot is a fungal disease. What else did we talk about? Talked about how about this one? Salmonella. Is salmonella caused by bacteria, fungi, virus, or a protist? Salmonella is food poisoning caused when you don't prepare food in a hygienic way. Often comes through chicken if you don't heat chicken properly. Good, lots of people saying bacteria there. Well done. Is caused by bacteria. Okay, this is working really well. There's some people still using the question and answer one. Use the chat if you can, but that's brilliant. Okay, so we've had a quick recap there. It's well worth looking back over any revision notes that you have or checking out BBC Bitesize if you want to know more about those diseases or have a little recap of those. Okay, so let's think about exam technique. So we get lots of different types of questions in exams, and I'm going to run through as many of those as I can today. So we're going to start with 1 or 2 mark questions, which are usually quite simple. They can be join the lines exercises or fill in the words. So in this case, we've got to draw a ring around the word. We're also going to have a look at slightly longer questions. We're going to have a look at how to go about answering questions with graphs in or tables. And we're going to have a look at slightly longer essay type questions and how you should answer those as well. So, 1 or 2 mark questions. Got a quick question that's come in on the question and answer session. What is algae? Again, sorry, so algae are different again. They're another type of organism. They don't cause disease, so we haven't looked at them in this module at all. You'll look at them a little bit more when you look at different families and how we classify different organisms as part of the course. Someone can't hear me as well. I'm not quite sure why that would be. That might be more of a technical issue. I know that my mic's working fine, so might be something different. Someone else has asked, what type of pathogen is corona? Corona is a virus, coronavirus. Okay, so 1 or 2 mark questions. The best thing to do with 1 or 2 mark questions is to read them carefully. They're usually directly testing your knowledge, so they are usually fairly simple to answer, and the trick is not to read them slightly wrong and waste that mark that you know already. So in order to combat that, I usually suggest that you underline the keywords if you can, or in some way highlight the keywords in the question. So for this question here, viruses and bacteria cause disease in humans. So we know that they're talking about viruses and bacteria and that they're causing disease. Part A, draw a ring around the correct word to complete the sentence. So we know we need to complete the sentence. Organisms that cause disease are called algae, pathogens, or vaccines. The correct answer there is pathogens. Pathogens are organisms that cause disease. That's the definition of them. It's 1 mark question, so that's all we would need to do for that question. So 1 or 2 mark questions are very simple, but it's important to read them properly. It is a shame to waste those marks if it's something that you need the answer to and that you just read wrong or answered slightly wrong. So let's look at another one. It's a bit of a larger example. Okay, so question 6 here, on paper. Lungworm is an infection. Lungworm can kill dogs. It's caused by a small worm. So we make sure that we read all of this description before we carry on at all. The diagram below shows the life cycle of the lungworm. So when we've got a diagram, make sure that you read each part of it thoroughly and make sure that you fully understand it before you go on to the question. So we've got 3 parts here. Part 1, lungworm reproduces in the dog and infects the lungs. The larvae, young stages of the worm, are coughed up from the lungs and swallowed. Stage 2, the worm larvae pass out in feces and are picked up by snails. So they've passed through the digestive system there, come out in feces, and they're picked up by snails. So we started in dogs, now we're in snails. Then, part 3, the dogs touch or eat the snails. And so then we return to the dog. It's a very interesting life cycle. It involves 2 different organisms that you wouldn't necessarily connect, but they obviously come into contact fairly often because lungworm is found in dogs fairly regularly, not all the time. The question that they've asked about this one, having had a good look at that diagram and the introduction to the question there, is what type of organism is represented by the snail in the life cycle of the lungworm. So the key words in there are type of organism, because that tells us the sort of answer that they want us to provide. And we know that they're talking about the snail within that diagram that we just talked through. They've asked us to tick 1 box, and the options that they've provided are fungus, parasite, protist, or vector. And it's a 1 mark question, so we need to tick the box for 1 mark. So, good, there's a couple of people saying vector there. And vector is the right answer. So, fungus we've talked about. A parasite is a type of organism that uses another organism to survive. So for example, a tick is a parasite because it uses the blood from organisms to survive. If you've ever had ticks on your dogs. A protist is a type of organism that we talked about as well. And then a vector, which is the correct answer here. A vector is an organism that carries a disease without getting it itself. So it passes it on to other things. So it's the same, it's how we also describe a mosquito when we're talking about malaria. They carry the disease between people, but they don't catch it themselves. Vector. Okay, any questions about 1 or 2 mark questions so far? Is this higher? This is foundation and higher level. Mostly we've concentrated on foundation level content. Okay, I think that covers all of the questions there. So let's move on to explain questions. So explain questions come up a lot. They tend to be 2 or more marks. And they're usually the ones that students find more difficult because they're asking you to interpret some data or to provide an explanation rather than just providing the knowledge that you have of that topic. So, got an example here of a 2-mark explain question. A key word in an explain question is always the word explain because it's the command word, and it's always worth underlining the command word in a question if you need to. Sometimes you'll have different ones, so usually you'll have explain, you might have interpret, you might have other examples of what they would want you to do. So it's worth knowing and making sure that you attach that to the number of marks. So here, some diseases can be cured by using antibiotics or prevented by vaccination. So they've used 2 of the things that we talked about last session for treating, curing, or preventing disease. For A, part 1, they've asked you to explain fully, so explain in detail, why antibiotics cannot be used to cure viral diseases. It's a 2-mark question. We need to provide 2 points or a key word. Make sure that we comply with that 2 marks. We've talked about this quite a lot, explaining why antibiotics cannot be used to cure viral diseases. It's something that's quite important to remember. Particularly at the moment in coronavirus is a viral disease. It cannot be cured by antibiotics. That's why we're working hard on a vaccination so that we can prevent it rather than treating it with pills. Okay, so I thought it'd be handy for you guys to see what a mark scheme looks like. So I've put a screenshot of the mark scheme for that question here. And I've also in red put the kind of worked answer at the bottom as well. So looking at this mark scheme, which is this section here, you can see that they've said that for 1 mark, you need to say that viruses live inside cells. There's a little 1 there, it's quite small. That means you'd get 1 mark for saying there. And so viruses are inaccessible to antibiotics because they're inside the host cell. So it's another mark there. They have said that you can allow drug/antibiotic used, and you are allowed would have to kill cell if you treated using antibiotics. So in full, I've written out kind of a model answer there. So viruses live inside host cells. Antibiotics target bacterial cells, so are unable to get to viruses without killing the host cell. Viruses are really clever. They use our own cells to shield themselves from any sort of treatment so that we can't treat them without killing our own cells. Okay. Got another one here. Another 2-mark question that follows on from the one that we had before. There's been a large increase in the populations of many antibiotic-resistant strains of bacteria in recent years. Explain why. So this is quite a big explanation. Antibiotic resistance, we talked about this, there was a whole flow chart behind it. For this question, it's 2 marks. So you need to make the 2 most important points that provide an explanation for why there's been a large increase in antibiotic-resistant bacteria in recent years. So again, I've put up the mark scheme so you can have a look at what exactly examiners are looking for with these types of question. So here is the mark scheme section here. So quite often on a mark scheme, they'll give students the benefit of doubt and they'll say, oh, any 2 from, or any 4 from, or any 5 from this list of reasons or list of things that you could mention. So don't always feel like examiners are looking for a specific thing. Sometimes they are, but sometimes they're looking for any of a number of things. So here they've put 3 bullet points and you needed 2. Let's underline that. So you can have any 2 from. For example, non-resistant strains are killed by antibiotics, as they should be. That's what we use antibiotics for. So there's less competition. The resistant bacteria have less competition against them, so they're more likely to survive, reproduce, and become a bigger population of resistant bacteria. And finally, overuse of antibiotics or antibiotics are prescribed for mild infections. And if no marks gained, allow 1 mark for people do not finish course of antibiotics. So that gives you a bit of an insight into what they're looking for there. So I've written a model answer here as well. The overuse of antibiotics to treat mild infections and then the patient not completely finishing the course of antibiotics means that antibiotic-resistant bacteria are able to occur and then increase as non-resistant strains are reduced by antibiotics, so there's less competition. So that's kind of brought in lots and lots of the different elements mentioned in that mark scheme there and put it into a complete sentence. It's important where you can with explain questions to answer in complete sentences if you can. Sometimes, particularly longer questions, it can be handy to use bullet points, but even so, those bullet points should still be written in complete sentences if you can. Even if they're short sentences, they should be complete sentences. Okay. Any questions about answering explain questions? We're going to look at longer ones, so 4 or 6 mark questions later, but for the moment, any particular questions? Looks as though we've had some tech problems. All sorted. Someone's asking if I can scroll up a little bit. That should be fine. Someone's just asked, do bacteria reproduce? The answer is yes, they do. They don't reproduce in the same way that we do. This is something that you'll learn as another part of the course and that you'll learn more about if you go on to do biology A level as well. So bacteria reproduce rather than sexual reproduction, which is what we use and what most animals use as well, and plants. Bacteria reproduce through something called binary fission. It allows them to reproduce very, very quickly, which is why they adapt very quickly and why you can see an increase in antibiotic-resistant bacteria very quickly if you're not careful. Okay, I've had a couple of people saying that they can't hear me. Can you let me know? If that's a problem at your end, my tutor. But for now, we'll carry on. So, questions interpreting graphs and tables. So, graphs and tables need quite a lot more reading into when you see those in an exam, and it's important to make sure that you do that properly before you even get onto the question. So let's work through how to do that. So here we've got a good table, lots of data from an experiment. So the table shows information about the development of a new sleeping pill. So we're talking about the development of drugs here. We talked about all the different tests that take place when they're developing drugs last time. We'll go through these in quite a few questions today. So the pill is for sleeping, so we know the function of it there. There's 4 rows: type of test or trial, preclinical, clinical phase 1, clinical phase 2, and clinical phase 3. Tested or trialed on cells, tissues, or animals. 20 to 100 healthy volunteers, 100 to 500 volunteer patients, 1,000 to 5,000 volunteer patients. Number of compounds tested. More than 10,000 compounds tested in the preclinical trial. 5 to 10 compounds tested in the clinical phase 1 trial. 2 to 3 compounds in the clinical phase 2 trial. And 1 pill in the clinical phase 3. So we've got a steady decrease there in the number of compounds tested as we move through those different trial phases. Time taken for test or trial in years. So 1 to 4 years for the first preclinical trials, another 2 to 4 years for clinical phase 1, another 1 to 3 years for clinical phase 2, and 2 to 4 years for clinical phase 3. So that's a lot of time there to develop a sleeping pill. Now we can move on, having had a good look at that table and look at the questions that they've asked about it. So what is the shortest time taken to develop a new sleeping pill? Okay, it's 1 mark. So the shortest time would be assuming that each of those clinical phases or preclinical phase took the shortest time it possibly could. So that would be 1 for the preclinical phase, 2 for clinical phase 1, another 1 year for clinical phase 2, and another 2 years for clinical phase 3. So 1 plus 2 plus 1 plus 2, so 3, 4, 6 years would be the shortest possible time to develop a new sleeping pill. We test all the drugs that we develop very rigorously, and it's not unusual for it to take 6 years to fully develop a drug to combat a disease. So that's why when we get something like we have at the moment, coronavirus, which is spreading very rapidly, it's affecting a lot of people. It is quite an aggressive virus, depending on the form that you catch. And we're having to work at double time to try and develop a vaccine or develop a way of combating that disease and make sure that it's safe. It would be foolish to miss out any of these stages, because it might mean that the drug either doesn't work properly, causes really awful side effects, or is toxic to people. So we need to go through all of these stages to make sure that a drug is safe to use. Okay, what is the range for the number of volunteers needed to complete all the clinical trials for the new sleeping pill? That's quite a complicated question for 1 mark. So they're looking for a range of numbers, and they're looking at the volunteers needed to complete all of the clinical trials, and it's 1 mark. Okay, so you could have, at the lowest range, you could have 20 plus 100 plus 1,000. So that would be 1,120 volunteers, if you had the lowest number. Or at the highest numbers, you'd have 100 plus 500 plus 5,000, so 5,600. And to get the range, you could either write out both of those numbers, so 1,120 to 5,600, or you could take 1,120 away from 5,600 and write down the answer to that one. I'll do that quickly. Use a calculator to make sure Be a shame to get it wrong when all of you probably have calculators in front of you, since you might have been expecting physics. 4,480 the example is the answer to that one. Excuse my terrible writing. Never have been very good at this. Okay, so there's a really good example of a table with some quite detailed information in it, and then a couple of fairly simple, but you also needed to think them through, 1-mark questions there. Okay. So let's move on to another question about graphs instead. Okay, so we might have to zoom in a little bit to look at the table. I wanted to get them both on one screen here because this involves you completing the graph from the table. So let's zoom in a bit. It might be a bit blurry, but we can work with that. Okay, I'll read it out as well. So the table below shows the number of people in the UK diagnosed with gonorrhea in different years. So, got the years down the left-hand side here, 2005 to 2013. And then we've got the number of people diagnosed with gonorrhea in thousands, and they're split into females and males. Use the data in the table to complete the graph below. The numbers for males have already been plotted, and only some of the numbers for females have been plotted. Okay, that seems simple enough. Let's have a look at the graph itself. I'm going to move over here. Okay, so it's important when you're looking at a graph to notice the axes. That's the first thing to do. Make sure that you know what the units are, you're familiar with the axes, and is there any patterns in the graph itself. So here on the x-axis, we have our year, which corresponds to the left-hand side of our table, 2005 to 2013. And then on the y-axis, we've got the number of people diagnosed with gonorrhea in thousands, which corresponds to our right-hand side 2 columns of our table. Both the graph and the table have the number of people diagnosed with gonorrhea in thousands, so we don't have to do any conversions there to make them fit. So they're literally asking us to put points on the graph. Okay, so the male line is the dotted line on the graph. We can see that if we're going to do a bit of data interpretation there, cases of gonorrhea plateau between 2005 and 2009 and then take a bit of an increase between 2009 and 2013. For females, we've got data plotted for 2005 and 2007. They stayed at 5,000 cases of gonorrhea. Then we can see from our table that in 2009 that goes up to 5.5. So we find 2009, travel up the graph, looked at our y-axis, and we can see that between 5 and 10 there are 5 lines. So 1 line for each number, so 5, 6, 7, 8, 9, 10. And we want 5.5, so we're going to plot this halfway between 5 and 6. Then in 2011, we have 6,000. So again, we'll pop this at the line above 5, be 6 there. And then 2013, we have 7.5. So we'll find our 5 line, count up 2, that will give us 7, and another half, 7.5. We can see on our key that the female line is a solid line. So what we'll do, and you'd be able to do this much better with ruler, would be to join up these lines here, these points here, and give ourselves a trend there. Okay, so that would be plotting the graph there. That's a good exercise in keeping cool, making sure that you read the table properly and making sure that you're reading the right units on the y-axis of the graph. Then we've got a question about describing the graph that we've just drawn. This is quite nice. You've already drawn the graph, so you're quite familiar with the data. But again, always worth making sure that you read these questions properly. Here, our command word is describe the patterns in the number of males and females with gonorrhea from 2005 to 2030. Use the data in the graph. This is really important. Not only is your command word describe, they've also told you explicitly to use the data in the graph, so make sure you use some of those numbers that you've just plotted or just noticed on the graph. So I've pulled up the mark scheme answer here again. I think it's really good for students to see mark schemes to get a good idea about what examiners are looking for. So you could have any three from, let's read them out and then we'll pop back to the graph and have a look at them on there as well. So you could have that levels of number of people with gonorrhea stayed fairly level up to 2009. We noted that when we first looked at the graph and we talked about the male line, and then we kind of noticed it when we were plotting the female line as well. They'll allow you to notice the very slight differences as well. That shows that you're looking at the data properly. There is a rise after 2009. You could have a mark for that. Males are always higher than females. The male dotted line was always higher than the female solid line. And males are rising faster than females as well. You could have any three of those for the full 3 marks. Let's go back to the graph. Here we are. There's my not very neat plotting there. I'm sure yours would be much neater in an exam. But we can see that, as we noticed before, and as we've just gone through in the answers, males always have higher cases of gonorrhea than females. We can see that males go up faster after 2009 when we noted that change, and that numbers do stay roughly the same between 2000 and 2009. There's a slight dip there in males and a slight increase in females, but not as much as there is after 2009. Good. Have a quick look at that one again. Now we're moving on to longer essay type questions. So, any questions about answering questions with graphs? Use the question and answer section. Can hydrochloric acid kill viruses? Okay, this is a question from the section before, I think. Hydrochloric acid can kill viruses if the virus makes it to the stomach. So we have hydrochloric acid in our stomachs, and we have mechanisms in our nose and in our airways, so our trachea and our bronchi and our lungs, to try and stop pathogens getting as far as the lungs and make sure that they get down to the stomach. So we talked about ciliated cells that have hairs, and we talked about goblet cells that give out mucus. So we have mucus that lines our airways. That's designed to catch pathogens, and then the ciliated cells with the hairs waft that mucus back up to our mouth, and we swallow it back down to our stomach, which is full of hydrochloric acid. This works very effectively most of the time, but occasionally we don't manage to waft all of the pathogen back up. And so we do get pathogens that affect our lungs, such as coronavirus. Coronavirus affects our lungs, our ability to breathe. And that's because our goblet cells and our ciliated cells just can't keep up with that pathogen and make sure that it all gets to the stomach. If it does, will it leave some burn marks on your skin? So hydrochloric acid we have in our stomachs. You certainly should not be using it on your skin. It's one of the first rules that we learn when you go into a lab. If you're dealing with acid, you make sure that you wear goggles, your lab coats and gloves. You don't get it on your bare skin. It will burn you. Can the coronavirus spread through air through droplets generated when an infected person coughs, sneezes, or speaks? Yes. That's the answer there. Usually with coughing or sneezing. That's why we're maintaining that 2 meter distance. Is the line a dot to dot or line of best fit? That's a really good question. So let's go back to the graph and have a look at the type of data that we've got. So we can see from the male dotted line that we have roughly straight lines between points. So that gives us an indication. But if we look at the data that we have in the graph, we've got an exact number for each of our points on our x-axis. We don't have any data for the years in between. So for example, we don't have any data for 2012. We don't know how many people had gonorrhea at that time. So for this case, you do need to join the dots. For this graph, it's not a line of best fit. There's a join the dots one because the data here it's not continuous. It doesn't include data for points in between the values that we've been given, so the years that we've been given. Okay. How do you know to draw a line of best fit? So we usually use lines of best fit when we have continuous data, or tends to be when you've got graphs with lots of values in them. So this is quite a difficult one to explain. Usually, if you are drawing a line of best fit, the question will say, drawn a line of best fit through these dots, or through this data plotted on this graph. And then they'll ask you to describe trends in the data, whether you think there might be a correlation. So is there an increase or a decrease? Usually lines of best fit are for continuous data rather than discrete data. So this is discrete data because we've got discrete points, discrete years. Usually lines of best fit are for continuous data when it's plotted on a graph. Okay, longer essay type questions. Oh, gone too far. Okay, so I've got 2 of these that we're going to have a look through. We've got 10 minutes left, so we're going to run through these quite quickly. I think we've answered most questions as we've gone along, so that should be okay. So 4 to 6 marks. Our first one, we'll underline the keywords as we've been doing all along. It's an explain question. How vaccination makes a person immune to a disease. So we're talking about vaccination and immunity. It's 4 marks. So generally for a question, you need to make as many points as there are marks for this type of question. So 4 points, 4 marks. I've pulled up the mark scheme again for you to have a look at. So these are the suggestions that they have given for those 4 marks. So the first one is that the virus or microorganism is introduced. That's the first mark. Notice that they've said ignore disease or germ. You don't introduce a disease, you introduce the pathogen, so the virus or the microorganism in a vaccine. You introduce it in a weakened form. The next stage is that vaccine stimulates the white cells or lymphocytes or leukocytes, which are the white blood cells in our immune system. We've called them white blood cells or lymphocytes. So far, either of those is fine. Some other exam boards teach leukocytes as well. There's something that you'll learn more about at A-level. You don't need to put that in now. Except B and T cells and ignore phagocytes. So the reason that they've said except B and T cells is that for higher level, you talk about B and T cells as part of the immune system. For foundation level, we don't talk about those. Ignore phagocytes. So if you remember, we talked about 2 different types of white blood cell. We talked about lymphocytes and phagocytes. Phagocytes engulf and destroy pathogens, but it's the lymphocytes that produce the antibodies that are important for vaccinations to work. So it needs to be the lymphocytes that we're talking about here to produce antibodies. Ignore antitoxins or antigens. You need to say antibodies, so be very specific with your language here. For the third mark, antibodies are made quickly on reinfection, or idea of memory cells, which again we talk about at the higher level but isn't needed for the foundation level. So here, again, you need to be quite specific with your language, but the whole point of a vaccination is that if you're reinfected with the disease, or with the pathogen, in order to cause the disease, then the antibodies are made very quickly when the body recognises that pathogen again. That means that the immune response is really quick. And that's how vaccinations work. So that's how you'd get the 4 marks there for vaccines. There is a cool graph for vaccinations that we talked about as well, which can help you understand this, and that you can look up on BBC Bitesize or any other good revision websites if you need to brush up on this. So here is a worked answer to that question. So you could say that a weakened form of the pathogen is injected into the body. That's our first mark. So 1. Stimulates the lymphocytes to produce antibodies that are complementary to the specific antigens on that pathogen. So we've gone into quite a lot of detail there, but we would get another mark for stimulating the lymphocytes and for producing antibodies. On reinfection, the antibodies are remade quickly to get rid of the pathogen. That'd be our 4th mark there. Okay, let's do another one. Longer essay type questions. This one is a 6-mark question. So this is more of a sort of essay type question. In 2014, the Ebola virus killed almost 8,000 people in Africa. Drug companies have developed a new drug to treat Ebola. Explain what testing must be done before this new drug can be used to treat people. Okay, big question. So when we looked at our table question earlier, we talked about the different types of testing that's used. So the different stages that take place while you're developing a new drug. Here is the mark scheme. It's worth having a good look at this slide if you can. So at the top here, they've given 4 levels of answers that you need to meet to get each of 2 marks. So let's explain that. For 0 marks, you could write no relevant content or not answer the question at all. For level 1, which is 1 to 2 marks, you can make 1 or 2 relevant points, but little linkage of points or logical reasoning between them. So they're looking for you to write a very short essay about this, making sure that you link your sentences and your explanations. So for level 2, for 3 to 4 marks, a partial answer with errors and ineffective reasoning or linkage. So if you've written a couple of explanations, but they are relevant, even if you put in a couple of errors, you can still achieve that 3 or 4 marks out of 6. So it's well worth writing complete sentences and giving these questions a go, even if you're not quite sure. For 5 to 6 marks, a clear, logical and coherent answer with no significant redundancy. That means that there's not much of the content that's not relevant. The student understands the process and links this to reasons for clinical trials. So that would be the best level you could achieve. Then they've given a list of indicative content. So they've said that preclinical trials of the new drug on cells, tissues, or live animals. So that's the preclinical stage of the testing before we move on to humans. To test toxicity, dosage, and efficacy. So whether the drug works, whether it's toxic, whether it has any side effects, and what dose we should use. Then you move on to clinical trials for testing on healthy volunteers at low doses, so that you can monitor for safety or side effects. And only then do you do trials to find the optimum dosage and test for efficacy, so whether it works at what dose. You can use placebos, which we talked about. So that's a bit like when you use a double-blind trial. So if you have two groups, you give each of them a pill. One of the pills is the drug, one of them is a placebo, so it doesn't do anything. But the patients in that group still feel like they've had a drug, so it avoids any mental side effects of taking a treatment. Random allocation of Ebola patients to groups, so that's talking about good science there. Make sure that you allocate people randomly, so no one knows who has the placebo or the new drug. And then you make sure that any results are peer-reviewed by other scientists to help prevent false claims. So you don't have to write all of that. That's an idea of what you could write. So I've written a model answer. So the first one is a not so good answer. Testing should be done on animals and people to see if there are harmful side effects of using a placebo. Okay, well that first sentence is right firstly, so we should do tests on animals and people, but you don't really need to see whether there's harmful side effects of using the placebo. The placebo is the blank drug that we give to people in a double-blind trial. Results can then be published after peer review and the drug can be used. Okay, I'd probably give that answer 3 marks. There were some inconsistencies, but they've clearly got the idea that you needed to do some testing, publish the results after peer review, and then you can use the drug. Answer 2 is a really good answer. So firstly, preclinical trials are conducted on tissues and animals to test toxicity, dosage, and efficacy. Secondly, clinical trials take place on human volunteers at low doses so you can monitor for harmful side effects. A placebo may be used that does not contain the new drug. Patients will be randomly assigned so they will not know if they're taking the drug or the placebo. The results will be peer reviewed so that no false claims are made. That would be a 6-mark answer there. So you notice I haven't mentioned everything that was in that bullet point list, but I have mentioned quite a few of them, enough for 6 marks. And I've linked all of my arguments together, all of my sentences together, to explain fully what testing must be done before the new drug can be used to treat people. Okay, we have 1 minute left. I'm going to have a quick run through and answer any pressing questions. I will pop back to the slide before, because I think you might want to have a better look at some of that content there. Can you write your answer in bullet points when writing your full mark answer? If you need to, then yes, but as I said before, I would still make sure that those bullet points are in full sentences. But if it helps you to describe a process, then you can. Can animals drop in the category of being vectors in respect of talking of coronavirus as they're carriers of the virus? I don't think we know enough to say that yet, to be honest. There's been lots of reports in the news of where coronavirus came from, and we suspect that it did jump from animals. The closest match has been to a virus found in bats, but we're not 100% sure of that as far as I know. I think everything is still very much up in the air, and scientists are working as hard as they can to work out what's happened there. Okay. Are leukocytes the same as lymphocytes? Not quite, they're slightly different. You learn more about those at A-level. Again, you don't need to know them for GCSE particularly. You need to know about lymphocytes, but not specifically about leukocytes. It's worth checking with your specification. Here we've talked particularly about AQA, but we've tried to tailor it so that it's roughly the same as other specifications as well. So it's worth just double checking if you aren't studying AQA. Why don't phagocytes produce antibodies? So phagocytes are cells that are purely there to engulf and destroy pathogens. So they're not, so we talked right at the beginning of GCSE Biology about how cells differentiate and become specialized for their function. So phagocytes aren't differentiated and specialized in order to produce antibodies. They're purely for engulfing and destroying bacteria.
I think that's all that we have time for today.
No problem.
Very much for an amazing session there, Charlotte. Everyone remember that all of our videos are uploaded onto our YouTube channel. So if you want to go over any bits, do feel free to go onto the channel to go to rewatch the video if necessary. Thank you very much for joining us today. Have a lovely afternoon. Keep safe, everyone. Bye.
Everyone.